Good morning, ladies and gentlemen. Thank you very much. I'm very grateful to the organizers who invited me to give a presentation and to present an overview on what we can learn from animal models in terms of changes in thyroid function during aging. So I don't want to preempt the next speakers, but just a couple of slides just to give a bit of context to the topic. So this study shows TSH distribution curve in a population study reported by Dr. Sirks and Holloway. And it basically shows that TSH levels increase when people get older. Studies reported by Dr. Mariotti in the 90s show that although T4 levels do not really change, T3 levels decline throughout life. And this has been consistently reported in many other studies as well. And together with an increase in reverse T3 concentrations, this suggests that peripheral metabolism of thyroid hormone is changed during aging. So from this slide we can conclude that thyroid state changes during aging. Some 90 years ago, Dr. Robertson reported on studies which he performed in mice where he administered desiccated thyroid extract. And what he noticed was a 15-week shorter survival when these mice got thyroid tissue. And this is quite significant on a lifespan of about 100 weeks. On the other hand, when rats were rendered hypothyroid, that resulted in an increase in lifespan of about 10 to 15%, also quite significant. And as a final example, I show you dwarf mice, which lack a pituitary transcription factor. So these animals are growth hormone deficient, these are hypothyroid, and they have a prolonged lifespan, as you can see. When only thyroxine is replaced, that markedly reduces lifespan. So from this slide we can conclude that also thyroid state in itself can affect lifespan. So I went through the literature to see at which level thyroid hormone could be affected, thyroid hormone regulation could be affected during aging. So either at the central level of the hypothalamus and pituitary, at the level of the thyroid gland, and at the level of the thyroid hormone target tissues. So to start with the central level. So this is data showing that circulating levels of T4 and T3 are decreased in old rats. However, TSH concentrations do not really change in the context of these lower T4 and T3 concentrations. And it's thought that is due to the fact that a type 2 deiodinase, which converts T4 to T3 in the pituitary, contributes to this observation. So in aging rats, TSH appears unaffected, while circulation T4 and T3 levels are lowered. So what about the thyroid gland? So this question has been addressed in the 50s, and this is a representative example shown, reported by Dr. Visser, basically showing that in old animals, in old rats, thyroid hormone secretion from the gland is diminished. And that nicely ties in with studies in humans also showing that thyroid hormone secretion from the gland was reduced. So this slide reminds you how thyroid hormone is regulated at the level of the T3 end organs. So thyroid hormone transporters ensure adequate uptake of thyroid hormone into cells. When thyroid hormone enters the cells, it can be either activated by the type 1 or type 2 deiodinase or inactivated by the type 3 deiodinase. And thyroid hormone action, as you know, is exerted when the active T3 binds its nuclear receptor. So in a study performed by Dr. Moradian, in old versus young rats, when they were injected with T3, uptake of T3 in some skeletal muscle and in brain was not different, while however in the liver T3 uptake was reduced. A couple of studies have addressed what happens with D1 activity in the liver. And as you see in the different studies, the type 1 deiodinase activity decreases in older animals. I couldn't find much what happens with the receptor during aging, but I would like to mention a very interesting study by the Aranda group showing that T3 can induce cellular senescence, but this effect is specifically mediated via the TR-beta isoform and not via the TR-alpha isoform. So if we summarize these studies so far, it shows that in the context of reduced T3, T4 concentrations in the aging animal, TSH levels appear to be unchanged. The thyroid gland appears to secrete less thyroid hormone into the circulation, and at the level of the end organs, at least in the liver, the type 1 deiodinase activity is reduced. However, actually there are a limited number of studies out there, and studies which reported on T3 target tissues mainly report on liver, and actually most of the studies are actually descriptive in nature, so the underlying mechanism which drives these changes is not really known. And this is conceivable because these aging studies are cumbersome, because they take time in themselves, and that also requires an investment of money. So if you take one step back and consider what aging actually is, then we might come up with a definition which is commonly used, and that defines aging as a time-dependent decline in cellular function, ultimately resulting in an increased vulnerability of death. And as such, it's the primary risk factor for many human diseases. A couple of years ago, some well-known aging scientists summarized what they believe to be the 9 hallmarks of aging. It should be realized, however, that not all parts of this circle, so not all 9 hallmarks, have equal weight. So actually it's genomic instability which is by far the most important contributor to aging. Well, all the other hallmarks are either consequences or responses to genomic instability. And if you realize that our genomic DNA is continuously damaged all day by either exogenous factors such as UV light or chemicals, or endogenous factors such as metabolism, which produces reactive oxygen species, that can result up to an astonishing amount of 100,000 lesions per cell per day. And that results in a great number and a great variety of DNA lesions which can all alter cellular function. So fortunately enough, our cells are equipped with mechanisms which repair DNA. So the consequences of DNA damage can be twofold. So if DNA damage results in a misreplication, actually resulting in an increase in cellular fitness, that drives cancer. While on the other hand, if DNA damage results in a block of transcription, that results in senescence and contributes to the aging phenotype. And the causative role of DNA damage in humans is well illustrated by patients who carry mutations in DNA repair pathways. So these patients suffer from progeroid syndromes, so they have an accelerated aging at a very young age. So various DNA repair deficient mouse models have been generated, and they faithfully mimic many of the features seen in these progeroid patients. But as you can see, these animal models also recapitulate many features of true aging. And therefore it has been proposed that these animal models can be used to study the normal process of aging as well. And that is kind of supported by this graph showing the liver transcriptome of a progeroid DNA repair deficient animal compared to normal aging, normal wild type aging animals. And as you can see, with increasing age, the similarity of the livers become more significant. So I would like to address this very important concept, which is called the survival or the adaptive stress response. So if DNA damage accumulates, resulting in the inability to transcribe DNA and in cell senescence, that contributes to the aging phenotype. And somewhere in this process, there is a trigger which elicits a so-called response, a so-called survival response, which includes an upregulation of antioxidant defenses and a downregulation of metabolism, of which the growth hormone IGF-1 axis is a prominent example. And here's the paradox, because usually in animals which have a perturbed growth hormone IGF-1 axis, such as IGF-1 receptor knockouts or dwarf mice or calorie-restricted mice, they show a prolonged lifespan. Well, however, in these proteroid animals, they do show a very short lifespan. And this is explained that these accelerated aging mice, and also in normal aging mice, that they employ a response to lower metabolism and thereby limiting the exposure to damaging molecules, thereby aiming to prevent further deterioration. And by doing so, the organism shifts its energy from growth and proliferation to preservation and maintenance. So we thought that it might be worthwhile to study these DNA repair deficient mice to better understand what occurs with thyroid hormone signaling during aging. So in the next couple of slides, I would like to share you some data we obtained in 2 different models and also on normal aging wild-type animals. So on the left side you see CSB XPA double knockout mice. So these animals lack basically 2 genes which are important in DNA repair. They have a severe phenotype, so they die in about 4 weeks, and they have many features of aging. For example, they show a severe kyphosis after 3 weeks, which is usually seen in 2 years. The other model, the ESCC1, can live up to 6 months, so is moderately affected. And here you do see fibroblasts. When exposed to UV light, these cells cannot handle and they die. And below I show, as part of the survival response, a lowering of the IGF-1 levels in both models. So first we looked at the liver at well-known T3 responsive genes, so D1 and SPOT14, and they were both markedly lowered in liver of both models. So then we studied the genome-wide expression profiles in normal aging animals of 130 weeks, in the 2 models, and also in hypothyroid animals. And as you can see, there's a subset of genes which is all regulated into the same direction. So this altogether shows that in livers of aging, of normal aging and accelerating aging, there's a distinct hypothyroid profile. So to investigate the cause thereof, we first studied serum levels of T3 and T4, and as you can see they were lowered in the CSB XPA, although nothing much happened really in this animal model. So then we looked at a thyroid gland, and when we examined these glands histologically, we could not find any abnormalities. So therefore we moved forward to look what happens at the T3 target tissues. So this shows you livers where we measured T4 and T3 concentrations, both lower in the accelerated aging model, and in particular T3 levels are discordantly low. And this is associated with a decrease in activity of the D1, of the type 1 deiodinase, and to our surprise there was an increase in the type 3 deiodinase. And this enzyme, as you may know, is usually expressed only during fetal life and can be reactivated in certain pathological states. And it's important to know that this upregulation already was seen at day 12 before these animals were severely ill or before they lost excessive body weight. So what happens in livers of normal aging mice? So in 2-year-old mice we kind of confirmed findings by previous studies showing a decrease in the type 1 deiodinase, and we found an increase in the type 3 deiodinase. And in particular, when we looked in very old animals, 130 weeks, there was a huge increase in the activity of the type 3 deiodinase. So in kidneys, basically a similar profile is seen, so a lowering of T4 and T3, accompanied by a lower D1 activity, but here the D3 activity didn't really change compared to the wild types. And in normal wild-type aging mice, there was a slight decrease in T3 levels, which was accompanied by an increase in D3 activity. So we then investigated the brain. Again, T4 and T3 levels were lower, but here it appears that T3 is less affected than T4 in the brain. And also the activities of the D2 and D3 enzymes were not different between the accelerated aging model and the wild types. And also in the other, in the ERCC1 mice, although T4 levels were lower when they were 18 weeks of age, T3 levels were maintained in the brains of these animals. So it appears that in the brains of these animals, thyroid hormone signaling appears to be preserved. So in muscle, I won't go into detail on all these figures, but it basically shows that both in heart and skeletal muscle, thyroid hormone signaling appears to be preserved. So, because we noted in the liver that both the type 1 deiodinase was decreased and the type 3 deiodinase was increased, both in normal aging mice as well as in DNA repair deficient mice, it was very likely that DNA damage was the underlying cause. So to test this hypothesis, we treated wild-type animals with a pro-oxidant, the DEHP called, which at a very low concentrations put the genome at a certain stress. And here you do see basically the same picture, so a low D1 and an increased D3. So this supports that both in normal as well as in accelerated aging, it's DNA damage that underlies these changes in the deiodinases in peripheral tissues. So if we sum up what others and we have found, we can draw a picture like this. So DNA damage results in aging, and that triggers kind of an overall stress response, so including an upregulation of antioxidant defenses, a downregulation of the growth hormone IGF-1 axis, and also a specific of a modulation of thyroid hormone regulation in a tissue-specific way. So in liver and kidney, T3 levels were low, which was accompanied by low D1 and an increased T3 activity, so resulting in diminished thyroid hormone signaling. While in other tissues like brain and heart and skeletal muscle, it appears that thyroid hormone signaling was preserved. So to conclude, what animal models have taught us is that at a central level it appears that TSH levels are unchanged in view of a decrease in circulation T4 and T3 levels. Also, the thyroid gland reduces its secretion of the hormone into the circulation. At a T3 T3 end organ tissues, there appears to be a tissue-specific response, so a suppressed thyroid hormone signaling in liver and kidney, and a preserved thyroid hormone signaling in brain and muscle. And this might be well part of the so-called survival response. DNA damage likely links aging and the changes observed in thyroid state, and I think that DNA repair deficient mice can well be used as a model to understand normal aging. I would like to thank a couple of people who have contributed to some studies I showed here, in particular Chantal Zevenbergen from our group and Sander Barnhorn from the Jan Hoeimakers group in our institution, a number of collaborators, and part of the work I showed here was supported by the American Thyroid Association. Thank you.